So let's start at the beginning, and the beginning is, what's the basis for the architecture of cells? And the answer is the actin cytoskeleton. It creates structures that allow cells to fine-tune their overall architecture. And so it always seemed very obvious to take after the actin cytoskeleton as a potential chemotherapy target. Why? Because the architecture of the cell is involved in regulating the proliferation of the cell, its ability to undergo cell division, and its ability to migrate, which underpins metastasis. Over 40 years, people have been developing anti-actin cytoskeleton drugs as potential treatments for cancer. The intrinsic problem has been that through this time, there has been one inescapable problem, and that is the actin filaments also control contraction of muscle. Your cardiac muscle, your heart, relies on the same system to beat. The same thing applies to the diaphragm. And hence, the dilemma has been, if you develop anti-actin cytoskeleton drugs, they stop the heart from beating, they stop the diaphragm from contracting, and hence the toxicity is simply unacceptable. We went back to the basics and asked the question, what are the core constituents of this actin cytoskeleton, and is there an alternative way of taking it apart? The actin cytoskeleton is involved in essentially every cellular process. If you look inside the structure of a cell, what you find is that there are filaments of actin all throughout the insides of the cell, involved in a myriad set of cellular processes. So how do you specialize what filaments do what jobs? And what we started to realize as we dissected the process was that in fact the actin filaments that are involved in building this architecture are not naked. That is, they don't consist just of actin. Many of them consist of two components. Shown here in this slide, there is the actin at the core part of the filament, but there is also tropomyosin, and tropomyosin is this spindly molecule that you can see in the slide that is overlaying the actin filament. Now, the very interesting feature of this is that the tropomyosin exists in many forms. So in our bodies, we make over 40 different types of tropomyosin. Why is that interesting? Well, if you think about the evolution of the architecture of cells, and you go way back in time to yeast, then you look at plants, if you look at the slime mold, dictyostelium, if you look at the flies, if you look at humans, what you notice is that evolution has made two types of decisions. In the case of all organisms, they all make actin. And if you look in the plant and you look in the slime mold, they make many different types of actin. And these actins are used to do different jobs. So it allows you to specialize the job with the type of actin that will do that job. But what you'll notice from this slide that is really quite extraordinary is that while the plants and the slime mold have made these decisions to make lots of different actins, that's not the case for us as humans or for the fly or even for yeast. What these organisms have done is they've chosen to combine the actin with the tropomyosin. And what you see, for example, in this slide is that the yeast makes two types of tropomyosin. The flies make over 10 different types of tropomyosin. And as humans, our bodies make over 40 different types. So what this tells us is that evolution has selected for the diversity of composition of the actin filaments. Some organisms have done this by just making lots of different types of actin, but other organisms, such as ourselves, have chosen to use a lot of different tropomyosins to fine-tune how their architecture works. I'm going to summarize 30 years worth of research activity in one slide. And what I'm going to tell you is that what we have learned over the last 30 years is that the tropomyosins are located in different types of actin filaments that do different jobs. The tropomyosins form largely homogeneous polymers with the actin, and they regulate the total amount of actin filament that's made in a cell at different locations. They regulate the interaction of the actin polymer with organizing proteins. They also regulate the interaction with myosin motors. As a result of that, they control the physiology of our bodies, and they also underpin the activity of cancer cells. Why do you build an actin cytoskeleton like this? Why do you create this homogeneous set of different architectural elements? Well, I think they're probably homogeneous because it means that along the entire length of a filament, the same thing is happening at every point. You get fidelity of function. And obviously what you also find is that the different tropomyosins are functionally unique. So it's like deciding I'm going to use a different building block to do a different job. And you've got multiple jobs you've got to achieve in your cells, and the way to do it is different building blocks, so you tailor-make the building block for each job. At a practical level, what does this really mean? It means that when you build actin filaments in your body, you take actin on the one hand, you form a polymer, but at the same time you do that, you're adding a specific tropomyosin onto that actin filament. And that tropomyosin is now going to determine who can interact with that polymer. And that will mean you'll get this very specific functional outcomes from each of these specific tropomyosins. You would predict, therefore, that different tropomyosins would have many, many different functional roles within our bodies. And over the last 15 years, it's become clear what those roles are. So, for example, the structure of your nerve cells is regulated by a specific class of tropomyosins. The structure of cancer cells is also dependent on a specific set of tropomyosins. And the one I'm going to draw your attention to that's in red on this slide is a tropomyosin called TPM 3.1, because that becomes the basis for our story, which you'll see in a minute. But in addition to this, there are tropomyosins that regulate the structure of membranes in your cells. Other tropomyosins regulate the ability of a cell to move. Other tropomyosins regulate the cell junctions between epithelial cells. The cystic fibrosis transmembrane receptor is maintained in the membrane by another specific set of tropomyosins. Muscle repair relies on one specific tropomyosin, and the contractility of fibrotic structures created by myofibroblasts is also regulated by a specific tropomyosin. In other words, our bodies have selected different tropomyosins to do very specific different jobs that we rely on so our whole body functions effectively. Now, what we realized as a result of our research was that there was a specific tropomyosin found in all cancer cells that turned out to be very important. Today it's called TPM 3.1, but its original historical name is TM5NM1. And what we know about this tropomyosin is that it's upregulated in all cancer cells. It's an integral component of the actin cytoskeleton of neuroblastoma, melanoma, and in fact, essentially all tumor cells. It regulates the stiffness of cancer cells. It's required for tumor cell growth and survival. It's essential for the viability of embryonic stem cells. And it's required for the regulation of cell proliferation by the MAP kinase pathway. All this information immediately indicates to us this is potentially a very useful target for cancer treatment. So what we decided to do was make drugs that would compromise this particular tropomyosin. And the way we did this was to take advantage of the fact that the way you form this continuous polymer of tropomyosin is you have to overlap each tropomyosin in turn, so it forms a continuous polymer. And the way it does it is very simple. One end of the tropomyosin looks like this. It has a splayed end. The other end of the tropomyosin has a pointy end. And it's actually designed so that one end can interact with the other end, and in this way it forms a continuous overlapping polymer along the whole length of the filament. This occurred to us to be the perfect place to start thinking about developing drugs that would compromise the formation of this continuous polymer and hence compromise function. Now, key piece of information for you is that tropomyosin itself has a very weak interaction with the actin polymer. The key is this overlap. As you build this continuous overlap between the different tropomyosins that are forming a continuous polymer, so the different molecules, each of them is coming together to form this overlap. When you do this, suddenly you get a much more stable structure. So what you're doing is you're cooperating. All of the building blocks are coming together to cooperate with each other to make a continuous linear polymer, and it becomes quite stable. So what we decided to do was we would develop drugs that would bind to the splayed end of the tropomyosin. It would sit in this end, and it would change the way the head and tail of the tropomyosins were able to interact. And in so doing, the aim was to try and compromise the stability of the filaments that you then make. What we came up with initially was a drug called TR100. And for us, the key was to demonstrate that this drug would in fact interact with the target tropomyosin and change the way it worked. And in the slide here, so the slide is a bit busy, but what I want you to understand is if you look at the uppermost left-hand corner, what we are measuring here is the way in a test tube that the actin, the tropomyosin, and the drug interact. Now, under normal conditions, if I take an actin filament that is just naked actin and I cause it to depolymerize, to fall apart, it will fall apart quite quickly. However, if you look at that upper left-hand panel, what you'll notice is that there are two curves. The black dots show you the rate at which naked actin falls apart by itself, and the open circles show you what happens if you put the tropomyosin onto the actin filament. And what you see is it falls apart much more slowly. In fact, it hardly falls apart at all. We then did the same experiment, but we included the drug. And what I want you to see from this graph is that the difference between actin alone and actin plus tropomyosin is completely abolished when the drug is present. In other words, the drug prevents the ability of the tropomyosin to protect the actin filament. And that turns out to be the absolute key property of the drug. Now, one way that you can then look at the impact of the drug in a cancer cell is you can ask the question, if I take a cancer cell and I expose it to the drug, does the actin start to fall apart? That is, does the architecture start to fall apart? And what we noticed was indeed that is exactly the case. Now, the way you do that experiment, and it's shown on the bottom left-hand panel, what we are measuring here is the stability of the actin filaments in a cell. And what you can see along the bottom is that as you increase the amount of the drug TR100, you decrease the amount of actin filament in the cells in a dose-dependent manner. So what that's telling us is that the drug increases the amount of what we call soluble actin. And in fact, we also noticed that the neuroblastoma cells were more sensitive than normal fibroblasts. And that's a good thing because it's starting to indicate we've got specificity as well. So the next thing to do was to take the TR100 drug and see if in fact it would impact on the growth of cancer in a model of cancer in a mouse. So the way to do this is to in fact grow the cancer in a mouse and ask the question, if we introduce the drug into that mouse, can we control tumour growth? So the next thing to do was to take the drug and ask the question, if you grow a cancer in a mouse and expose the mouse to the drug, can you control the rate of growth of the tumour cells? So we did this using both melanoma and neuroblastoma. If you look in panel A, what you can see is that if we exposed the mouse growing the B16 melanoma and then exposed to the drug, what you see is the drug significantly impacts the rate of growth of the melanoma. In panel C, we did exactly the same experiment using a human melanoma-derived cell line. And then in panel D, did the same experiment growing neuroblastoma. In all cases, what we find is that the TR100 drug has the capacity to inhibit the growth of the tumour. So what that gives us is proof of concept that in fact the drug has the capacity to have effect against the growth of both melanoma and neuroblastoma. Now the question of biggest concern was, up until now, no anti-actin cytoskeleton drug has been able to progress through animal models because of the toxicity, because of the impact on the heart and the diaphragm. So we were most concerned to ask the question, was there any impact on the heart in these animals that had been treated with the TR100? And what you can see here is a series of experiments that were run to ask the question, was there any impact on the heart of these animals in which the drug had reduced tumour growth? What you can see in panel A is it's a picture of the hearts from control animals and from those who were subjected to the treatment with TR100. And what we found is at the gross level of the morphology of the heart, there was no impact. If we looked in panel B, we were measuring a protein that is released when the heart is compromised, and that protein then goes into your blood. And so we could just do a quick measurement of this protein in the blood of the animals that were treated with the drug and ask the question, was there any indication of heart damage? To which the answer is there was absolutely no indication of heart damage, which was fantastic. We also measured the weights of the hearts, and there was no impact on the weights of the hearts. What we now know about the tropomyosins is they regulate actin filament function. They do it in a number of different ways. They control the structure of the filaments, they define what motor proteins can interact with the filaments, and they control the stability of the filaments. But I think what's most exciting is that the tropomyosins clearly provide therapeutic opportunity. On the one hand, we've worked out how to drug them, how to turn them into a drug target. Cancer cells clearly depend on TPM 3.1 for survival. The anti-tropomyosins which target TPM 3.1 are now very promising anti-cancer agents. Now we know that the different tropomyosins are required for a range of different functions in the body, and in fact different tropomyosins associated with different functions. So now we can think of anti-tropomyosin agents which target other tropomyosins as potentially providing additional therapeutic potential for other functions within the body. Okay, so I want to acknowledge the people who've been involved in this project, and in particular I want to highlight Dr. Justine Stain, who was the team leader who was involved in the development of these drugs and the trialling of these drugs in the animal models. I also want to highlight Teresa Bonello. Teresa was a PhD student with us who worked out the protein chemistry that gave us an insight into how the drugs work. We have many, many collaborative partners throughout the world who've been involved in helping us traverse this very difficult path. We've been funded by the Australian National Health and Medical Research Council for much of the basic science that we have done. Finally, I want to thank the Kids Cancer Project. The Kids Cancer Project has made it possible for us to develop the therapeutics. They are focused on developing new therapies for childhood cancer, and in particular for neuroblastoma. And I want to particularly thank them and their founder, Cole Reynolds, who really have played a pivotal role in allowing us to take the basic science down a path towards the treatment of childhood cancer. Thank you very much.